it's always good to have a come to Northeast. So now I'm come to my talk. Before going to the main talk, I would just like to tell you that I have been working in the area of cancer biology and radiation biology. And we have developed, we have brought two molecules of interest from the as a radioprotector or radiomodulators, and then we have brought we have another utilization of this benzimidazoles we have done, that we have made them as a antibiotic development molecule, and we have taken it up to the preclinical level. So we have been doing a lot of things from chemistry to biology, signaling, cancer signaling, radiation signaling, and and pharmacokinetics, pharmacodynamics in our laboratory. So with all these things, I will come to the DNA damage and its repair and relation to the human diseases. Let us talk about it. That what are we? We are actually made up of approximately 50 trillion cells, and then we all know that there is a cell, there is a nucleus, there is a chromosome. And if you look into all those things are made up of DNA. So DNA is our genomic, entity, and it is one of the most important entity. And whenever we talk, whether we take it a nerve cell, cancer cell, or the heart cell, it starts from the DNA. So if we talk about the DNA, that is the molecule of life, and each cell almost have the 46 human chromosomes, 2 million DNA, 3 billion DNA subunits, 80,000 gene codes for proteins that perform all life functions. So DNA is a very important part of our, body, and it is made up of ATCG. I don't have to go in all those details, but if you look into the central dogma of the life, so you can see that everything revolves around the DNA, and we all know that there is a DNA, and then after the transcription there is an mRNA is formed, and then mRNA gets translated into the protein. And if you look into that, there are so many Nobel Prizes which have been related to the DNA, and that is 1962, 2006, and very recently the DNA damage and DNA repair was shared by, Nobel Prize was shared by 2015 by three chemists, and that was actually, you can say that they were given their due in the 2015, and they have this DNA repair pathway has solved so many problems related to the diseases, and actually told us, it clarified us that how these damages are occurring. And then finally you can see that even in the translation also, when the protein is formed, that also fetched a Nobel Prize, that is called as the 1972 Nobel Prize. So now the question arises, what are the agents that damage DNA? If you are in Delhi, I think we all have a damaged DNA. So there is so much of pollution. It is a beautiful weather here. You cannot imagine there what is happening. But if I just summarize here, UV light, ionizing radiation. I work with ionizing radiation. Smoking, a lot of inflammation because of the different reason, air pollution, environment damage, and now there are a lot of DNA damages are occurring, and on that DNA damages further, epigenetic changes are also occurring because of the urbanization also. So a lot of studies are being done on the DNA damage and how these DNA damages are leading to the epigenetic changes also. So these are also a certain new correlations are being found. And very recently I was visiting Colombo in a meeting of International Council for Scientific Union, and we have found that there are a lot of epigenetic changes have occurred in the South Asia Pacific region, and which was actually related to the DNA damage which could not be repaired properly. So DNA damage is a very important thing, and as it is written here that the thousands of DNA damage event occur every day in our cells, and it has to be repaired, and it has to be repaired efficiently. So that is the one word, and and there should not be any repair fault there. And if it is a repair fault, then that will cause a mutation, or this is called as a evolving term mutation can occur in the daughter cell. So if we talk about the DNA damage responses, so whatever DNA damages occur, so we always say all people who work with the DNA damage agents or the DNA, they say that the single strand DNA damage can be repairable, but if it is a double DNA damage, it cannot be repairable. So if you are talking about say single cell DNA damage or the double strand breaks, you can always say that but the most of the cases it has been observed that when I practically work on the radiation, that if I observe say 10 single strand breaks, I already observed 100 DNA strands breaks. So telling it that the single strand breaks only convert into the double strand breaks, we do observe double strand breaks also directly. And if you try to say cells have gone. So if you are working with say a double strand break or the single strand break, you can always say that the single strand breaks. So there are because of the single strand break, I'll come to the why this damage occur and what are the damage sensors there. But there are 3 different repair pathways which have been read clearly. That was the base excision repair, nucleotide excision repair, and then the mismatch repair. And there was another say repair methodology which comes actually into the say between the nucleotide base excision repair and base excision repair, which is called as the homologous recombination repair and the non-homologous end joining. So these are the 3 thing, and those 3 changes actually fetch the Nobel Prizes to 3 gentlemen. And if you look into the say these 3 gentlemen who got Nobel Prize, they were chemists. That was the intrinsic fragility of DNA, Thomas Lindell, Paul Modrich, and Ajit Sankar. So more or less, and they say if you listen their Nobel lecture, they all of them end their lecture with that the life exists, so DNA must repair. So whatever DNA damage is there, that should and I will go one by one to their studies. So if you look at the Lindell, I have included these slides because I wanted to say motivate some of the young students, young researchers here. So Lindell actually talked about the say he was born in Stockholm, and he has largely worked about the base excision repair, and he is a person who was largely in the born and brought up in United Kingdom. Then the second person, Modrich, he was the person who worked on the nucleotide excision repair, and he was also got Nobel Prize, and before that also he was quite a lot acclaimed person. And then Sankar was talked about the mismatch repair, and he also worked almost 40 years on the mismatch repair, and in his lecture he says that when he lost the hope, then he got the Nobel Prize. So with all these things, now if we talk about that what happens when there is a DNA damage. Whenever there is a DNA damage, there are certain damage sensors are there, and those damage sensors are recruited at the site of the DNA damage. Apoptosis happens, and the cell cycle transitions also occur. And then if I go to the molecular level, so what are the typical proteins or the genes which are affected by the DNA damage? So as soon as the DNA damage occurs, there is a replication stress, and ATM and ATR, these are the 2 kinases which are recruited there, and few other say important protein, RAD9, RAD1, a plethora of say genes and proteins proteins are recruited there, and they affect the cell cycle checkpoint, and those are the CHK1, CHK2, and these CHK1, CHK2 activation actually stall the CDK1, and that causes the cell cycle arrest, so that the cell cycle is delayed, so that the repair should be done in a proper way rather than hastily, so that because the wrong DNA repair will end up in the mutation. And then there is an activation of the, say, MDM2, and MDM2 is in a loop pathway with the P53, which stops the P53 expression, and that is how it causes the apoptosis. And there are certain DNA repair genes which are being activated, BRCA1, NBS, CABL, and there are so many others DNA repair genes which are recruited, and they start the repair, but repair has to be correct. There should not be any wrong repair should occur. So if we talk about the ATM and ATR, ATM, ATR are the two particular, regulators. These are the kinases, and these are called as the master kinases, because they actually meant that how the G1 arrest will be done. And the G1 arrest has been done through the activation of P53, and leading to the MDM2 activation, GAD45 alpha, P21, which once again stops the expression of the cyclin D1, cyclin E and cyclin D1, which causes the G1M G1 arrest, and the conversion of the G1 to M is delays. The another pathway which is being activated by the ATM ATR is the S1 phase, S phase delay. That is the preparation of the DNA replication is delayed here. And then the another third thing is the G2 phase arrest occur, and that is also leaded by the ATM and ATR. So that is why we call them ATM and ATR. We call them as a checkpoint regulator. So when we say that there are cell cycle checkpoint responses occur in the response of the DNA damage, so we can start it from the G2M checkpoint, or you can start it from here, that there is a gap of the growth phase, then there is a DNA replication phase, then G2 phase, that is called as the growth phase 2, then M is the cell division phase, and these are the cell cycle checkpoint. So these are the, as I've already told you, and in order to reduce my time, I would not explain it here, but I would say that these cell cycle checkpoints are being activated, and the delay in these checkpoints are occur. Conversion of the G2 to M is reduced so that the repair can be done properly. The response to DNA damage will be different depending on the cell cycle status. So the cell may be in any other phase. It may be G1 phase, G2 phase, M phase, or S phase. So depending on that, the cell cycle changes occur. So the activation or the checkpoint delays occurs. So now what are the need for the DNA repair? DNA Paul does a great job, but introduces errors in about 1 in 10 to power 7 nucleotides added, which it does not correct. So most mistakes and damage corrected, 99%, leaving just a few, only 1 into 10 to power 9 errors are left. Repair of non-replication related damage to the DNA must also be a priority for the cell. These defects also will prevent translation and duplication of the DNA, which leads cells to die. Again, any error to changes to the DNA become mutations, which are the permanent changes left in the DNA. So if I look into the Thomas Lindell, he talked about the base excision repair. So what is a base excision repair? So actually what happened, that DNA is a labile molecule, and it can be damaged by the, say, from the where the nucleoside bond is there. It can also be damaged by the and the damages can be done at the nucleic acid bases, and the damage also can be done by the at the different parts of the nucleic acid bases. So you call it hydrolytic attack, then oxidative damage, or there is an alkylation occurs also. An alkylation also modify the, for example, it modifies the different nucleic acid bases to different nucleic acid bases. So that kind of a DNA lability causes the depurination, and depurination is always talked in terms of the guanine. So the base excision repair, what happens here, that because of the say certain modification which I talked here, what there is, I have taken one particular example examples for the students that what happened that if you have a say RNA, in that RNA, if uracil DNA glycosylase comes and talks and sits here, and the uracils comes out of it, and that is how the base excision occur. And why base excision is occurring here? Because the glycosylase recognizes that there is a kind of a modification has occurred there, and that is how it comes there and remove that particular base from there, so that the repair can be done properly. So a particular base is being repaired here. And how it has happened actually, that the deoxycytosine have got methylated and it has converted to the deoxyuracil, and deoxyuracil, you know, there's nothing, and so then the enzyme recognizes the deoxyuracil and removes it from there in order to do the repair pathway. So this is the kind of lesions which are created. And if you look into the lesions, if you think that the lesions are are very small, you can see that if you see the 3 into 10 to power 9 base pair genome, you can find that the hydrolysis, say in the 3 into 10 to power 9 base pair genome of double strand DNA, you can see after 24 hours at 37 degree, these many hydrolysis, oxidations, and the non-enzymatic methylation can be observed. So there is a high rate of DNA damage occurs in the body, and it is sometimes by the external, chemical regions, and it also occurs normally also inside the body. So the high rate of DNA decay implies that the repair mechanism must exist and are continually active under physiological conditions. So the first one which Linda gave us the reasons was actually the base excision repair. So this is again given here that there is a cytosine which can easily lose an amino group forming a base called uracil. There is a uracil has come, now glycosylase will come, remove the particular base from here, and another couple of enzymes will come here, and then they will say polymerases will come here, then they fill up the place, they will bring back the cytosine, and then ligase will seal whatever is the whatever is the nick was happened here because of it. So then you get back the same from where you have started. But if there is a mismatch, if there is a proper repair has not happened, then it ends up in the mutation. Similarly, the second person, that is the Aziz Sankar, he talked about the nucleotide excision repair. That is the another pathway of the repair. And what he said actually, that because of the UV radiation, because of the radiation, what happened, that the two thymine, they come together and they make a dimer. And that dimer is being recognized by the certain enzymes, and it has been observed. And this is actually his paper from where I have taken this particular picture. So it talks about that there is an UV radiation occur, a thymine dimer is there, and almost a 10 to 12 base pair of the nucleic acid oligonucleotide is removed from here. Then again DNA polymerase comes here and fills up, and DNA ligase seals up to give you back the normal DNA. So this is called as the nucleotide excision repair mechanism. And the beautiful thing is about the nucleotide excision repair is that it is having a different pathway in the E. coli, and it has a different pathway in the humans. I'm not going into the detail of this, but the third one is the Paul Modrich, who was the third person who received the Nobel Prize in 2015, and that talks about the DNA mismatch repair. So what is the mismatch repair? The mismatch talks about that you have they say that there is when DNA is copied during cell division, mismatched nucleotides are sometimes incorporated into the new strand. Out of 1,000 such mistakes, mismatch repair fixes all but one. So there are 3 enzymes which are important, that is the MUT-H, MUT-S, MUT-L, which actually participate in the repairing of it. And what they do, that they remove the whole mismatch from there. From where the mismatch has started, they go and bind there, and they remove the faulty copy from there, and they put a proper copy there, and that is how the mismatch repair is occurred. So with all these things, now I will go, because my talk says that the human repair and the diseases. So I will just take 2 more minutes. So it is like say many of the DNA damages have causes a lot of diseases. So these are the diseases. More than the cancer, there are so many syndromes which are occurring because of these DNA damages. DNA repair. So these are the certain, syndromes which occur because of the DNA damage. These are the few. Ataxia, is one of the most important one. Then we have the next one. I'm not going into the mechanism of it. Xeroderma Pigmentosum is also one of the syndrome which occurs because of this, and this is the picture of the person who is suffering with the Xeroderma Pigmentosum. Then there are, another DNA repair disease is the dry cot thyroid dystrophy. This also occur because of the faulty repair in the DNA. Then this is another which occurs in the pediatric one, that is Cockayne syndrome. Then HNPCC syndrome. So HNPCC, this is called as this occurs, this is a colorectal cancer, and which also is found because of the mismatch mismatch in the DNA. And at the last, the most important one is the cancer. And as all of us know that if there is a DNA damage, and if there is a proper cancer therapy is given, and the tumor suppressor gene is still working, and the proto-oncogene is not activated, then you get a proper repair to the DNA damage, and there is no cancer. But if there is an error-prone DNA repair occur, that turns into the mutations or the rearrangements and activation of the oncogene and the deactivation of the tumor suppressor gene, causing the cancer. So with all these things, I would like to finish my talk because I think I've finished my time. Thomas Lindell has said at the end of his talk that scientific research is not based on vacuum, it is built on the work of your predecessors. So these are the acknowledgements. I've taken it from the official Nobel Prize site, some other sites. So this is all about this, and if I have any questions, I can answer.